Good afternoon students, this is your Prasanna Ma'am from NEET World, and here we are to discuss the advanced questionnaire on molecular basis of inheritance. So after looking into the NEET previous year questions in our 2 sessions previously we have done. So from those 2 sessions we have got to know that the questions which are raised from this chapter are mostly statement based questions, assertion reason type of questions, match the following type of questions, which most importantly are from the liners of the NCERT itself. So keeping that in our mind, we are targeting NEET 2026 and framed out certain set of questionnaire. Let's get started and exercise them. So first question is on the nucleoside. A nucleoside is formed through an N-glycosidic linkage by joining what? Just a second. Nucleoside means what? Nucleoside is nothing but sugar and phosphate group. Okay. So sugar and nitrogen base, sorry. Sugar and nitrogen base, we will call it as what? Nucleoside. Fine. Sugar is a pentose sugar, right ma? Sugar is a pentose sugar. So first carbon of this pentose sugar will be adjoined with nitrogen base. This nitrogen base can be a purine or a pyrimidine. So sugar and nitrogen base are associated by what bond? N-glycosidic bond. N-glycosidic bond. Fine. So this is by N-glycosidic bond. The nucleoside is formed through an N-glycosidic linkage by joining sugar and nitrogenous base. So what is the right answer over here ma? Pentose sugar and nitrogenous base. Option 3 will be the right answer. Next question you look into. Yes. Refer to the given figure of the nucleosome and select the option that correctly identifies the part A, B and C. Okay. A, B and C. This is a direct diagram of your NCERT wherein this is from what? Nucleosome model. This A is what ma? Histone octamer. This is what? Histone octamer. Histone octamer with 4 proteins of 2 sets. What are they? H2A, H2B, H3 and H4. So this is a histone octamer. To this histone octamer which is positively charged, the negatively charged DNA is wrapped around this. As such, DNA packaging happens. So the negatively charged DNA is wrapped around the positively charged histone octamer. Negatively charged is what? C. DNA is wrapped around this octamer, right? So C is DNA, and the DNA is connecting 2 histone octamers through a linker protein, that is B. Okay, B is what? Histone 1, H1 protein, that is linker DNA. Okay. Next, so here if you see, A is what? Histone octamer, B is H1 histone protein and C is DNA. Which option is right ma? Option B is the right answer. A is histone octamer, B is H1 histone and C is DNA. Clear, right? Next question. Assertion and reason type of question, very important, look into it. The sugar phosphate backbone of 2 chains in DNA double helix show anti-parallel polarity. This is from DNA double helical model. Okay, double helical model of DNA. This point is taken. The sugar and phosphate backbone of 2 side chains, they are anti-parallel to each other. Assertion is true. In reason, if you see, the phosphodiester bond in one strand go from 3' carbon of one nucleotide to 5' carbon of the adjacent nucleotide. All the time the linkage happens in between 2 nucleotides. In between 2 nucleotides, how the linkage happens ma? By phosphodiester bond. At 5th carbon, phosphate group will be attached, and at 3rd carbon, OH group is there, right? So this OH group is connected to the phosphate group of this phosphoric acid, phosphate group, by what bond? Phosphodiester bond. Phosphodiester bond. Fine. Again this phosphate group is connected to 5th carbon, right? Fine ma. So this linkage is what? Phosphodiester linkage between 3rd carbon of one nucleotide and 5th carbon of the next nucleotide of same chain. But then in reason what it is given? The phosphodiester bond in one strand go from 3 prime carbon of one nucleotide to 5 prime carbon of the adjacent nucleotide. No, it is to the next nucleotide of same strand, whereas those complementarity strands go vice versa. Never two strands in the double helical DNA are not complementary, they are anti-parallel to each other. So yes, reason is wrong and assertion is true over here. So which option you will be choosing, Vita? A is true and R is false. Option C will be the right answer. Option C is the right answer. Next question. Yeah, look at it. The genetic transformation experiments on Diplococcus pneumonia gave first undoubted evidence that what is the evidence that is given out from the Griffith's experiment on Diplococcus of transformation principle, right? That DNA is the genetic material, DNA is made of nucleotides, no, chromosome, no, there is no information about chromosomes and there is no point about RNA that is dealt in the transformation principle. So in transformation principle, which is given by Frederick Griffith, after experimenting through Diplococcus pneumonia, he said that there is some transforming principle that is transferred from heat killed strain to the R strain bacteria, which is making it virulent, okay. But then it is unequivocally proved that DNA is genetic material by whom? Hershey and Chase, who gave the experimental evidence for transforming principle, fine, which is given by Frederick Griffith. So here what will be the right answer, beta? Option A. Next question you look into, ma. Yeah, just a second. Yeah, during analysis of DNA of an organism having 5386 nucleotides, it was found out that adenine is 29%, guanine is 17%, cytosine is 32%, thymine is 22%. Considering Chargaff's rule, it can be concluded that. So what can we conclude considering Chargaff's rule? According to Chargaff's rule, in any double-stranded DNA, be it circular or linear, the nitrogen bases, number of purines will be equal to number of pyrimidines, and number of adenine equal to thymine, number of guanine is equal to cytosine. This is what said from the Chargaff's rule. But then here in the question, the amount or the percentage of adenine is not equal to thymine, the percentage of guanine is not equal to cytosine. So with this only we can confirm that this is not what? Double-stranded DNA. This is not at all a double-stranded DNA, be it linear or circular, no. This is a what? A single-stranded DNA. This is a single-stranded DNA. Option C is the right answer. Next question, this one. Look into it, beta. RNA is less stable genetic material as compared to DNA. True. The reason for this is, what is the reason for this? In RNA, second position has OH group while DNA does not. Yes, second position of pentose sugar, that is ribose. Second position of ribose sugar if you take, at second position, free OH group is present. This free OH group is the main reason for making RNA unstable and reactive. And the second reason is RNA is also known as catalytic. Yes, RNA is catalytic, example is ribozyme, okay. This is also catalytic and reactive. RNA has uracil in the place of thymine. Thymine in DNA will add additional stability to DNA, whereas absence of thymine and presence of uracil as nitrogen base in RNA is also one of the reasons for its instability. So all these reasons are correct only. In that case, what option we have to choose? More option D is the right answer. Next, assertion reason type of question. Assertion: DNA has evolved from RNA with chemical modifications that make it more stable. True. This we have learned from what? RNA world. Okay, RNA world from molecular basis. The point is very clear that DNA has evolved from RNA. Okay, DNA has evolved from RNA. In reason, if you see, RNA being catalytic was reactive and hence it is unstable. Both the points are true only. Okay, RNA is catalytic, example is ribozyme. This is it is reactive and thus it is unstable. But nowhere these 2 points are correlated, right? But then both are true. In that case, what option we have to choose, ma? Both assertion and reason are true, but reason is not the correct explanation of assertion. Option B will be the right answer. Next question you look into. Yeah. E. coli with completely radioactive DNA was allowed to replicate in the non-radioactive medium for 2 generations. The percentage of bacteria with radioactive DNA is. This question is from experiment of Michelson and Stahl. Okay, this question is from Michelson and Stahl's experiment. Michelson and Stahl's experiment. Michelson and Stahl's experiment clearly says what? It is an experimental proof for semi-conservative method of DNA replication. So, here in question what is he saying with a E. coli with complete radioactive DNA. Radioactive DNA is what? 2 strands are N15 only. Complete radioactive DNA was allowed to replicate in non-radioactive medium. Non-radioactive medium is what? N14 medium. This is allowed to incubate or replicate in N14 medium for 2 generations. 2 generations means 40 minutes. Okay, for 2 generations this allowed to replicate. The percentage of bacteria with radioactive DNA is. What is the percentage of bacteria after 2 generations? First we will see after 1 generation. We can see N15, N14 and again N15, N14. After 2nd generation. Okay, all this will be the parental strands and the 2nd generation is also incubated the same non-radioactive medium. So, what we will be getting beta? 2 hybrid strands, as 2 N15 are there, we will get 2 hybrids and 2 light strands are there, 2 hybrids and 2 light. 2 hybrid strands and 2 light strands. So, the percentage of bacteria with radioactive DNA. With radioactive DNA, what will be the percentage of bacteria? 2. That is half, 50%, right? Amongst the total, it is half, right? So, 50%. So, what will be the right answer over here, ma? Option B is the right answer. Next question you look into. Transcription requires which of the following enzymes? So, in transcription, if you see, for replication though, we need DNA dependent DNA polymerases, because the parental strand is DNA and the new strand to be synthesized is DNA. So, it is DNA dependent DNA polymerase. For transcription, if you see, the strand is what? DNA strand. Using the DNA strand, RNA strand is being synthesized. Okay. So, parental strand is DNA and the strand which is synthesized is RNA. So, in this case, what is the enzyme that is involved in the transcription? DNA dependent RNA polymerase, and RNA is being synthesized from polymerized from 5 prime to 3 prime direction. So, it is what? DNA dependent RNA polymerase. You can also call it as DNA based RNA polymerase. Option C will be the right answer. Next question you see, ma? Yeah. List 1 and list 2. Match the list 1 and list 2. Select the correct answer using code given below. Okay. First, ligase. Ligase, if you see, ligase will join the short segments of DNA. DNA ligase is the last enzyme which is used in DNA replication, which is used to bind what? Okazaki fragments, okay, in the lagging strand. So, ligase joins the short segments of the DNA. This is correct. Second is DNA polymerase, it will not cut the DNA at specific DNA sequence. What is that enzyme which will cut the specific DNA sequence? DNA is cut at the specific locations by endonucleases, okay, not by RNA polymerase. RNA polymerase is keenly involved in synthesizing a new DNA strand. Sorry, DNA polymerase is keenly involved in the synthesis of new DNA strand. Helicase, if you see, helicase will break the hydrogen bonds between the complementary pairs during DNA replication. True. Helicase, DNA helicase is the first enzyme which is starting the DNA replication. First it will cleave the double helical structure of the DNA by breaking what? Hydrogen bonds between two nitrogen bases of two strands of DNA. So here what are the correct combinations of the statements beta? 1 and 3 are correct, 2 is incorrect. So which option we have to go through? Option D will be the right answer. Next question you look into, ma. Yeah, methyl guanosine triphosphate is added at 5' end of heteronuclear RNA in a process of what? So methyl guanosine is added at 5' end. This is a post-transcriptional event which occurs inside the nucleus of eukaryotic cells. Okay, this 5' capping where methyl guanosine is added, this is called as capping. Second post-transcriptional event is what? Tailing at 3' end, polyadenylation occurs. And third event is what? Splicing, which is mediated by SNRPs, where introns will be excised and exons will be joined. Okay, introns which are inactive sequences will be excised and exons which are expressed sequences will be joined. These three are what? Post-transcriptional events which occurs only in eukaryotic cells inside the nucleus. Remember the location, that is very important. So with respect to this question, methyl guanosine triphosphate added at 5' end, this process is called as what? 5' capping. Next question, assertion reason type of question. In assertion, eukaryotic mRNA requires post-transcription processing for the formation of functional mRNA. Once again, eukaryotic mRNA requires post-transcription processing for the formation of functional mRNA. True, this is true. Okay, just now we have discussed those post-transcriptional events. In reason, if you see, eukaryotic transcripts possess extra non-functional segments called as introns. No, in post-transcriptional event only introns will be excised or lysed. Okay, so non-functional segments will not be present in the functional mRNA. So in this case, assertion is true and reason is false. Option C will be the right answer. Next question, beta. Yeah, which of the following statements about the genetic code is correct? Which of the following statements about the genetic code is correct? Genetic code is a triplet. Yes, it is universal. Ambiguous. No, it is not ambiguous. It is non-ambiguous. It is a triplet, universally accepted. One genetic code is universal. Okay, it is same in all the species. Of course, there are few exceptions with respect to mitochondria organelle might be and protozoans might be. There are very few exceptions, but then genetic code is universal. And then non-ambiguous. Every code, one codon is specific for one amino acid, that is non-ambiguous. Okay, and degenerate. One amino acid can have many codons. One amino acid, if you take for example leucine has 6 codons. Leucine amino acid has 6 codons. Phenylalanine has 4 codons as such. Okay, one amino acid has multiple codons. Multiple codons. Fine. So B is correct here. Right, ma? So this is the proper description about genetic code. Okay. Remember, genetic code is non-ambiguous and degenerate. Next. This you see, anticodon is a base triplet on. Okay, anticodon and codon. This concept comes during translation. Okay, codon is the one which is present in the mRNA. In mRNA, codon is present. Genetic codon is present. The code which is taken by the tRNA which sits on the larger subunit of the ribosome at the P site will copy this codon, and the code which is copied by the tRNA is called as anticodon. So tRNA complementary to a base sequence of rRNA, wrong. tRNA which is complementary to the base sequence of mRNA is called as what? Anticodon. tRNA which is complementary to the base sequence of mRNA is called as anticodon. Clear, right? So, option C is right over here. Next question, ma. Just a second. Just a second. Yeah, look at this question. Match the components of lactoperon of E. coli and find out the correct combination. If you have to see PYQs, many at times the questions asked on the lactoperon, very important model. So, here if you see, first will be what, ma? Promoter region. To promoter region always RNA polymerase will bind, remember. Next, followed to that operator region. To operator region always repressor will come and bind, and this is followed by structural genes, that is Z, Y and A. Okay. So, here the components of lactoperon in E. coli and find out the correct combination. Structural gene will code for what? Enzyme, be it Z, Y, A. Z codes for beta galactosidase, Y for permease and A for transacetylase. So, structural gene will code for what? They codes from enzyme proteins. So, A will be S. Operator region, to the operator region, this will be the binding site for repressor protein, that is P. Operator region is the binding site for repressor protein. Next, promoter gene is the binding site. Promoter gene is the binding site for RNA polymerase, that is T. Regulator gene will code for what? Repressor protein. Regulator gene will code for repressor protein, that is Q. Okay. So, here A is S, B is P, C T and D Q. Option C will be the right answer. Next question. In the lactoperon, the genes A, I, Y and Z code for what? Okay, important. A codes for what, ma? Transacetylase. A codes for transacetylase, I codes for repressor protein, and Y codes for permease and Z codes for beta galactosidase, that is option D. Next question. Which is incorrect about DNA polymorphism? The two alleles of a chromosome. What is DNA polymorphism first of all? Variations in the DNA or the variants which are observed in DNA is said to be DNA polymorphism, like variable number tandem repeats or the repeated DNA sequences that are seen in DNA, that includes under what? DNA polymorphism. So, here which statement is incorrect about DNA polymorphism is the question. The two alleles of a chromosome contains different copy number of VNTR. Okay, two alleles of a chromosome contains different copy number of VNTR. Two chromosomes contain different copies, no? Polymorphisms are inherited, yes, polymorphisms are inherited, and different chromosomes contain different copies of VNTRs. Last, if you see, DNA from different tissues from an individual show different degree of polymorphism. No, DNA from different tissues from an individual, from a single individual, shows same degree of DNA polymorphism, not different. So, what is incorrect over here? Option D. Okay. Next. Fine. And here if you see, match column 1 and column 2. Choose the correct combination from the options. Frederick Griffith. Okay. Frederick Griffith has worked out on Streptococcus pneumonia, which is option 4. Hershey and Chase has worked out on what? Bacteriophage, which is option 2. Michelson and Stahl's experiment on Escherichia coli. Taylor et al. He has given the experimental evidence on the eukaryotic cells. Okay, experimental evidence that DNA is a genetic material. Hershey and Chase has worked out on bacteriophage. Taylor et al. Has worked out on faba beans, that is option 3. So, here if you see, 1 is 4 and 2 will be 2, 3 is 1 and 4 is 3. Option B is the right answer. Next question. Select the correct statement. RNA polymerase 1 transcribes rRNA, true. RNA polymerase 2 transcribes snRNA, wrong. RNA polymerase 2 transcribes hnRNA, that is heteronuclear RNA. Next, RNA polymerase 3 transcribes hnRNA, wrong. RNA polymerase 3 will transcribe tRNA, 5S RNA and snRNA. Fine. RNA polymerase 2 will transcribe hnRNA. So, here option 1 and option 4 are correct. Okay, 1 and 4 will be correct. Option A. Next question. Select incorrect statement. Okay, incorrect statement. 6 codons do not code for any amino acid. No. Codon reading of mRNA is continuous. True. 3 codons function as stop codons. Yes. Not 6. 6 codons do not code for any amino acid. No, only 3 codons do not code for any amino acid. These are stop codons. Okay. So, stop codons can be what? UAA, UAG and UGA. Clear? Next, the initiator codon is AUG, codes for methionine. True, right? This is the dual nature of this codon. AUG is the initiator codon, at the same time this codes for methionine. And A alone is incorrect. Yeah, A alone is incorrect. 2 is right, 3 is right and 4 is also right. Yeah. So, A alone is incorrect. Option A will be the right answer. Select the incorrect statement. A alone is incorrect is correct. 2, 3, 4 are incorrect. This is wrong statement, right? So, yes, option A will be the right answer. Yes, so this is the last question of today's session. In our next class, we will be exercising the questions, NEET previous year questions on microbes and human welfare. Okay? So, that is all for today's session, students. Thank you.